Welcome back to another episode of Genetics Unsealed. I'm your host, Philip Delegate, and in this episode, I'll be delving into the captivating genome of one of the world's favorite beverages, coffee, and shedding light on two fundamental biological processes, mitosis and meiosis. Mitosis, a critical form of cellular division in eukaryotic organisms, plays a pivotal role in repair, growth, and asexual reproduction. The process consists of four distinct phases: prophase, metaphase, anaphase, and telophase. Before mitosis occurs, we have interphase. Interphase comprises of three substages: G1, S, and G2. In G1, the cell essentially continues its normal functions, preparing for the upcoming division. During the S phase, the cell undergoes DNA which is something I'll go through in another episode. It ensures that each chromosome duplicates itself, ensuring that when the cell divides, both resulting cells will possess identical genetic material. And it's essentially the cell's way of making sure it has all the necessary instructions for the forthcoming split. Following the S phase, the cell enters G2. Here it continues to prepare for division by following and checking any errors in the newly synthesized DNA. Interphase is at the backstage of theatrical performance, where the chromosomes get ready, genetic material is checked and rechecked, and the cell, or the stage, is set for the grand act of mitosis. The first phase, prophase. Picture the cell. The nuclear envelope dissolves and the chromatin condenses into visible structures called chromosomes. These chromosomes, now distinct and identifiable, become key players in the subsequent stages. Moving on to metaphase, the chromosomes gracefully align themselves along the cell's equator. This alignment is no random feat. It ensures that each daughter cell receives an equal and identical share of the genetic information. The spindle fibers, like puppeteers, orchestrate this symmetrical arrangement. The stage then transitions to anaphase, the phase where the reaction takes place. The spindle fibers contract, separating the paired chromosomes at the centromeres. Thereafter, the chromatins journey towards opposite ends of the cell, ensuring that each new cell inherits a complete and identical set of chromosomes. The final phase, telophase. At this stage, the cell undergoes a remarkable transformation. The chromosomes, having reached their respective destinations, begin to decondense back into chromatin. Simultaneously, a new nuclear envelope forms around each set of chromosomes, creating two distinct nuclei within the cell, and thus the mitotic performance concludes. What started as a single cell culminates in the creation of two identical daughter cells, each with the same genetic information as the original. This meticulous process of cellular division is fundamental for growth, repair, and asexual reproduction in eukaryotic organisms. Transitioning from the singular orchestration of mitosis, our focus now shifts to the intricate duet of meiosis, a nuanced choreography that commands the stage in a profound narrative of sexual reproduction. Meiosis is a complex process essential for the creation of gametes, sperm and egg cells in animals, each harboring its unique genetic constitution. This process unfolds through two sequential phases: meiosis 1, also known as reduction division, and meiosis 2. The commencement of meiosis 1 is marked by prophase 1. When talking about meiosis, always mention a number to differentiate it to mitosis. A stage Prophase 1 is a stage imbued with heightened anticipation. Chromosomes undergo condensation, and homologous chromosomes, those possessing genes for the same traits but with slight differences, engage in synapsis, which is the fusion of chromosome pairs. This embrace facilitates a mesmerizing exchange of genetic material, identified as crossing over. Such genetic exchange bequeaths a richness of diversity to the ensuing gametes. Progressing to metaphase 1, the paired chromosomes elegantly array themselves along the cellular equator in bivalence. Unlike the ordered alignment observed in mitosis, these pairs assume a random disposition, further amplifying the intricate genetic combinations destined for the resulting gametes. The delicate hand or spindle fibers orchestrates their placement within with meticulous precision. Anaphase 1 signifies the separation of the homologous chromosome pairs, wherein each chromatid gracefully journeys towards one of the daughter cells. This measured segregation ensures that each resultant cell inherits a distinctive and diverse genetic complement. Telophase 1 demarcates the denouement of the inaugural act, accompanied by a brief intermission. The nuclear envelope reestablishes its enclosure around the chromosomes, creating two nuclei within the cell, poised for the subsequent act in meiosis 2. Meiosis 2 echoes the stages of mitosis, with a unique cadence. The two daughter cells from meiosis 1 undergo individual mitotic divisions, culminating in the birth of four haploid cells. These entities, now gametes, bear half the chromosomal bank of the mother cell. The final curtain descends on meiosis, revealing four distinct and genetically diverse gametes, each endowed with the potential to partake in fertilization, orchestrating the emergence of new life. Exploring the intricate genetics of Arabica coffee, a recent study by Nature Communications unveils the challenges and triumphs hidden within its large and complex genome. Led by Michael Morgan and Gabriel Di Gaspero, the research presents a more comprehensive genome sequence of Arabica coffee, shedding light on its unique history of hybridization with Robusta coffee. Arabica coffee, constituting 60% of global production, emerged from a fusion of Robusta coffee ancestors and another closely related species, Coffea eugenioides. Its hybridization bestowed Arabica with its renowned flavor and a challenging and intricate genome. Various technologies allowed for detailed analysis of Arabica's chromosomes, revealing differences contributed by progenitor species. Notably, variations in genes associated with caffeine biosynthesis were uncovered, adding to the allure of coffee. Surprisingly, genome analysis showed a low level of genetic diversity in Arabica coffee, with diversity spiking in specific cultivars due to chromosomal abnormalities and genetic segments from the Robusta Arabica hybrid, the Timor hybrid. The Timor hybrid, a genetic mixture, has become the parental line for many modern cultivars, blending disease resistance from Robusta with Arabica's unique flavor. The authors propose that Arabica coffee's genetic diversity is crucial for its commercial success, offering potential insights for developing new varieties with desirable traits. That's all for today. Thanks for listening to my podcast, Genetics Unsealed. Till the next time, signing off.